Hello all, welcome back. Today let us start the unit on molecular plant pathology. So the disease causing agents which are found in plants are similar to those that are seen in animals or humans. That is, either they can be microorganisms like viruses or bacteria, fungi, protozoa or nematodes, or the disease causing agent could be an unfavorable environmental condition, like either the lack of nutrients or the excess of nutrients, or it could be lack or excess of moisture, light, etc. So either of these could be the disease causing agents. Now, when do we call a plant as diseased? It is called as diseased when its physiological functions such as cell division, cell differentiation, photosynthesis, translocation of the photosynthates from the leaf to different parts of the plant, transpiration, etc. All of these physiological functions, when they are affected and when the plant fails to give its expected yield, that is when the plant is said to be diseased. And a manifestation of a disease. Manifestation is when a plant is said to be diseased, how is it shown? So the manifestation of the disease depends on which tissue the pathogen is going to attack and various disease causing factors. So to sum up, we can say that for a disease to develop, there has to be interaction of three components. Those three components are what are shown in this triangle. So the three components are the pathogen, the environment and the host. Now these three are represented in the form of this triangle, which is called as the disease triangle. The disease triangle has three sides. Each of the sides represents one factor that is important or crucial for the manifestation of a disease or for a disease to occur in a plant. One of the sides is the pathogen. So when we say pathogen, it means it is the totality or the sum of the virulence of the pathogen, how virulent it is, how much it is able to affect a host, or the abundance of a pathogen, that is the microbial load, how much of that pathogen is present near its host, etc. So anything with respect to the pathogen, if we talk about with respect to the organism, that comes on this side of the triangle, one side of the triangle. Another side of the triangle is the host. Host is the sum of conditions which favor the susceptibility. How susceptible, how sensitive is the host to that particular pathogen? How sensitive is this particular cultivar of the host? There are different cultivars that can be found or different strains that can be found in every plant. How susceptible is this cultivar to a particular pathogen? So, how susceptible it is, that is what comes in the second side of the triangle, that is the host. The third side of the triangle is represented by the environmental factors or the environment. Now this is a sum of or a total of conditions which favor the disease. It is possible that some diseases are favored during the rainy season. There are other diseases which are favored during the season of flowering. So there are several environmental factors like temperature, pH, so many factors which contribute to a disease actually occurring. So that is the third side, that is the environment. The area that is covered by this triangle, by the disease triangle, is the amount of disease. So if any one of the sides is more, say we have the environment side being more, even then the area increases and the amount of disease will be more. Or if the pathogen side is bigger, again the amount of disease will be more. So this triangle represents what amount of disease or how much disease can happen in a particular plant. On the contrary, if any side is zero, if any of these factors or components is zero, then the disease cannot occur. So there has to be a proper cooperation between these three, a proper interaction, a perfect interaction between these three components for the disease to occur in a plant. This is called as the disease triangle. Now when we talk about the disease cycle, it is the chain of events which leads to development of a disease, or basically it refers to the appearance of symptoms, the development of symptoms, and finally the perpetuation of a disease as a function of the pathogen. So the pathogen acting over there and showing all of the disease parts is called as the disease cycle or the development of the disease, in other words. Now you can see over here I have drawn the disease cycle. So let us start with the dormant period. So every pathogen will undergo a dormant period or a period when it is not active but it is alive. This period is called as perennation or it is called as overwintering. So the organism will survive in the dormant period, and when the favorable conditions arise, it forms the primary inoculum. Now in some cases, because we have already finished the unit on plant pathogens, you will be knowing that in some cases the primary inoculum is ready. It is already in the dormant period and it is ready to be spread, or it is ready to be disseminated. But in other cases, one particular spore has to sporulate, or it has to germinate, or there has to be some change. Say a telutospore has to give rise to a basidiospore, then that forms as the primary inoculum. So dormant period is when we are having the organism being inactive, and primary inoculum is when it forms the inoculum for dissemination. Now dissemination is the spread of the inoculum. This can be through air, water, or through transplant material like grafts, or it can be through insects. So every pathogen has its own methods of dissemination or spread. Now when the inoculum is disseminated, it goes and falls on a substratum, or it goes and falls on a particular pathogen where it is incubated. So if it is falling in the soil, it is incubated over there, wherein it recognizes a host. So from the incubation, from the time it is falling on a suitable substratum, it has to recognize the host and attach to the host. Now recognition of the host is by various factors which we will be studying. So the organism has to locate the host, recognize it, and attach to it, or adhesion. Attachment or adhesion is the next step, wherein the organism has to attach to the particular host. So it is not enough if it just recognizes it, it has to even attach to that particular cell. After the attachment, the organism has to penetrate inside the host. Now from the penetration, the infection starts. So once the organism has penetrated, it has got inside the host plant, now it establishes the infection. After the infection has been established, now for the infection to be established, the main factor is that the plant has to be susceptible. Also, in some cases the symptoms are shown immediately, whereas in other cases it can lead to a latent infection. So it depends on the plant, it depends on the pathogen, it depends on the environmental conditions, all of it, all the components of the disease triangle will decide whether the infection is going to be latent or it is going to be shown immediately. Also, it depends on the pathogen and the host whether the infection will be localized to a tissue or whether it is going to be systemic. Localized is where the organism enters through a part of the plant and stays in that part. It enters through the leaf, it divides in the leaf, it invades the leaf, it ruins the entire leaf, it might spread to the other leaves, but it will stick to the leaves. That is called as localized infection. Systemic is where the organism doesn't stop with the leaves alone or that particular leaf. It is going to spread through the entire plant. That is called as a systemic infection. So the infection, this is going to be decided by the pathogen and the host. Now once the infection has been established, the organism starts invading or invasion. Invasion is where it spreads to different parts of the plant. It starts colonizing, or it grows and there is reproduction of the pathogen. That is called as colonization, which leads to symptom development. So when we have a foreign body, a pathogen multiplying so abundantly inside the plant, automatically symptoms start developing. Now once the symptoms develop, there can be two ways that the flow of the disease cycle can occur. So from the symptom development stage, it can happen that the organism produces the overwintering stage of that particular pathogen. That means the organism decides to undergo the unfavorable conditions. So it has to form a structure that is going to help in perennation, that is going to help in overwintering. So production of overwintering stage is one of the processes that can happen. If the conditions are favorable, or if the organism is in a setup, in an environment where the conditions are favorable for spread of the disease, then after the symptoms develop, that pathogen starts disseminating from there. Now that is called as the secondary inoculum. So the organism disseminates or spreads from the plant that it has initially affected, and that is called as a secondary inoculum, and the cycle is repeated. So it disseminates to the neighboring plant. In the neighboring plant, again we have host recognition, attachment, penetration, and the entire cycle will repeat. This is called as disease cycle. So in the disease cycle, we will know how the organism is going to manifest itself or how the disease is going to develop. This is called as the disease cycle of every pathogen. Now for the university syllabus, if you see your subunit on molecular plant pathology, that is unit 5, if you see the syllabus, we have various topics. Some of them have been divided into seminars. So I would just like to present an overview how the flow of the chapter is going to be. and what are the different topics that have been given in your syllabus, so that it is easy for you to understand when either I take the class or when the seminars are done. So when we talk about the disease cycle, the first thing that, like I told you earlier, is the organism has to interact with the host. So the first part is the host-parasite interactions. Now these host-parasite interactions have to lead to an infection. So what are the different host-parasite interactions that can lead to an infection? The first host-parasite interaction that leads to an infection is germination. So once the spore has germinated, the spore germination is the first thing. The spore has to germinate, or if it is a parasitic plant, then the seeds of that plant have to germinate. Once they have germinated, we now have something called as a pre-penetration phase. Pre-penetration phase is basically location of the host. So yes, it has fallen on the soil, it knows there is a host nearby, but it has to locate it so that it can go on to the next step, that is adhesion. So from pre-penetration, we now have adhesion. The organism has to adhere, it has to attach onto the host cell. And from adhesion, we go on to the last stage, that is penetration and entry into the host. So all of these come under host-parasite interactions. This is what we will be starting with. Once the organism has entered inside and it has established an infection, definitely it is going to affect the host cell, or it is going to affect the host. It is going to affect the behavior of the host. The plant physiology is going to be affected. This is going to be studied, or we will be studying this under alteration of host cell behavior. Now what are the different processes that are altered? It can be either one process or all or many processes, like photosynthesis, translocation, or absorption of water, transpiration, respiration, or permeability of the cell membrane. All of these processes get altered, leading to wilting, or it can affect the seed production, it can affect the inflorescence. Different processes can be affected. Now once the host cell behavior has affected, the plant is not going to sit quiet. The plant is going to have its own. So what are the different mechanisms of disease establishment? Now the organism usually uses mechanical force to penetrate, but inside, once it goes inside, the manifestation of the disease or the progress of the disease is mainly due to chemical reactions. So we have production of enzymes, there are a production of phytotoxins, we have growth regulators. So toxins, enzymes, growth regulators are going to be produced by the organism, and the organism also produces some polysaccharides. All of these are going to alter the host cell behavior or establish the disease. So we will be studying these components which are produced by the pathogen. Now like I said before, the plant will not sit quietly. It does have some defense mechanisms. So the resistance mechanism in plants can be either the structural defense mechanism, or it can be the biochemical defense mechanism. Structural defense mechanism is structural details which are already there. It could be either pre-existing, or it could be some structure that is developed in response to the attack by the pathogen. Biochemical also similarly could be a pre-existing biochemical defense, or it could be some biochemical defense that is developed post the infection. So it occurs due to signaling mechanisms, which is also a seminar topic for you. These post-infection biochemical defense mechanisms include phenolics, generation of nitric oxide, callose development, phytoalexins, HRGPs, PR proteins, that is pathogenesis related proteins, which lead to systemic resistance. So all of those topics are going to be dealt with under resistance mechanism of plants. Now there are some genes which are present which help in providing resistance. So either providing resistance or making the plant susceptible. So under the genetics of plant disease, we will be studying about elicitors, which are substances produced by the pathogen of biotic origin, which induce the defense response in the host. So elicitors which are produced by pathogen, and the presence of R and R genes. Capital R is the resistant gene, small r is the susceptibility gene, which contributes to the disease development. Followed by this, we will be studying about molecular mechanisms of disease diagnosis, which includes serological tests, fatty acid profiling, or protein analysis, PCR, etc. There are different mechanisms by which you can diagnose a disease, apart from finding out the symptoms, which is a main method, but there are a lot of molecular methods also which help in diagnosing a disease. And finally, you will be studying about the transgenic approaches which are present for protecting crops. So this is what the unit comprises of. I'll be sharing with you the different seminar topics which are there under these, but this overview should help you. Thank you.